So just let's start with the last session of today's event, in which a successful scientific project will be presented with the aim to link science to business and wider society. After each presentation, there will be a discussion guided by technology transfer professionals, Jure Vindišar from the National Institute of Biology, Tomaž Lutman from the Jožef Stefan Institute, and me, Vojka Žunić, from the National Institute of Chemistry. So the aim of this discussion will be to identify future steps needed to achieve higher TRLs of the presented scientific work. But I also encourage you in the audience to join the discussion by directing questions and comments via chat box or in person by raising your hand. So we can start with the first project, which will be presented by Professor Dr. Mojca Jenštrle-Sever from the University Medical Center of Ljubljana. Professor Jenštrle-Sever, please.
Lepo pozdravljeni, samo da začnem deliti ekran. A se vidi?
Not yet.
Okay.
Zdaj pa? Samo če date še...
Ja, je v redu, ok. Sem Mojca Jenštrle-Sever, zdravnica iz kliničnega centra, endokrinologinja, in vam danes želim povedati eno zgodbo o tem, kako lahko črevo nagovarja možgane in v bistvu kako zelo sta povezana dva pomembna sistema, fiziološka sistema vseh živih bitij, in to je po eni strani prehranjevanje in po drugi strani reprodukcija. To bo zgodba o hormonu glukagon podoben peptid 1, ki nastaja v črevesju in se začne sproščati, ko pridejo hranila v črevo. Potem pa začne spodbujati inzulin na trebušni slinavki in pa, ker smo dobili hranilo v telo, takoj začne signalizirati tudi možganom, predvsem našim centrom za apetit, da se ta naš apetit začne zaustavljati, hkrati pa v manjši meri začne tudi upočasnjevati praznjenje želodca. Zdaj, ta naravni hormon se zelo hitro razgradi, je pa ta hormon, zdaj teh hormonov v tej komunikaciji črevo možgani je ogromno, ampak ravno ta GLP-1 hormon je pa zanimiv zato, ker je v bistvu z manipulacijo prišel v pomembno farmakološko uporabo. Tako da so danes analogi njegovih receptorjev za GLP-1 nekako predstavljajo ključne igralce, ki bojo krojili to zaenkrat zgubljeno bitko z debelostjo. Imamo že registrirani dve zdravili, GLP-1 agonistov, in sicer liraglutid in semaglutid, in zlasti zadnji zelo uspešno uspe z vplivom na doživljanje našega apetita zmanjšati telesno težo, tudi tja do 15%. Tako da to so novi igralci v desetletju in naslednjih desetletjih v tej bitki proti debelosti. Potem imamo pa po drugi strani naše ožje področje, s katerim se pa res vsak dan srečujemo endokrinologi, to je pa sindrom policističnih jajčnikov. Ta sindrom prizadene okrog 5 do 10% žensk v reproduktivni dobi in v bistvu predstavlja eno izmed najpogostejših endokrinoloških motenj. Gre za zelo kompleksen hormonski koktejl, ključna motnja pa je nekako signalizacija med možgani in med jajčniki. Tukaj, če si predstavljamo zdaj hipotalamos kot nekega menedžerja, se ta motnja začne na tem nivoju v izločanju in pozitivnosti hormona, ki ga imenujemo GNRH, in potem se ta motena signalizacija takoj prenaša na našega trenerja, če si predstavljamo zdaj to ekipo v tej osi kot nogometno moštvo na področju hipofize, ki izloča LH in FSH. Ker je signalizacija z vrha motena, strani GNRH hormona, pride tudi do neravnovesja v izločanju hormonov hipofize. Tako da je LH hormon se izloča v višjih koncentracijah, kar normalno, kar pa vpliva potem na nogometne igralce v tej osi, se pravi na te tarčne organe, predvsem na tekalne celice v jajčnikih in vodi v nenormalno povečano izločanje moških hormonov. Hkrati pa se tukaj znotraj jajčnikov vmešavajo še drugi hormoni, kot je antimillerjev hormon, ki zavira pa zorenje jajčnih foliklov. Tako da te ženske v bistvu trpijo po eni strani zaradi znakov prekomernega izločanja moških hormonov, kar pomeni, da se akne, poraščenost, izpadanje las vleče preko adolescence v odraslo dobo, in po drugi strani v ginekološke motnje, v motnje menstrualnega ciklusa. Temu kompleksnemu hormonskemu koktejlu pa se pridružuje tudi inzulinska rezistenca, ki je specifična za ta sindrom. Se pravi, da je deloma neodvisna od debelosti, jo pa seveda debelost močno poslabša, hkrati je pa selektivna. Nekateri tarčni organi so podvrženi inzulinski rezistenci, nekateri tarčni organi, predvsem jajčniki in hipofiza, pa ostajajo dobro občutljivi na inzulin. In naša raziskovalna skupina se je že pred več kot 10 leti začela spraševati, kaj bi naredili ti agonisti, GLP-1 agonisti pri tem sindromu. Saj več kot 50% žensk podvržena debelosti, kot smo poudarjali, inzulinska rezistenca pa je temu sindromu intrinzična. Zanimalo nas je, ali bo to rezultiralo samo v redukciji telesne teže in izboljšanju inzulinske rezistence, ali se bo zgodilo še kaj drugega, kaj novega. In začeli smo opažati, da s tem zdravljenjem izboljšamo menstrualne cikluse teh žensk, kar je deloma razložljivo za redukcijo telesne teže, ne pa povsem. In potem smo pa naredili še študijo z bolj usmerjenimi reproduktivnimi outcomi ter ugotavljali, da so izhodi reproduktivnega zdravljenja pri teh ženskah, ki so zelo težko zanosile, boljši pri uporabi GLP-1 agonistov. Tako da smo si začeli spraševati, ali ni morda potrebno v to enačbo GLP-1 vsi prinesti novih parametrov, se pravi te vsi hipotalamus, hipofiza, jajčniki, morda celo endometri. In smo naredili tak zelo poglobljen narativni pregled literature, več kot 1000 člankov smo pogledali, tako predkliničnih kot kliničnih, in ugotavljali v bistvu zelo zanimive stvari. Da se ta hormon iz črevesja, GLP-1, res kar konkretno in direktno vpleta v reproduktivno os na vseh nivojih te osi. Začne se to vpletanje že na področju hipotalamusa, ki smo ga identificirali kot osnovno motnjo pri sindromu policističnih jajčnikov, potem direktno vpliva tudi na hipofizo, direktno vpliva na jajčnike in tudi na endometri, kar bi potencialno lahko pri reproduktivnih postopkih izboljšali to reproduktivnost. Deloma preko zmanjševanja vnetja in deloma preko popravljanja motenih signalizacij na tem področju. Tako da je bilo zelo zanimivo prinesti v to zelo široko GLP-1 super os, kjer so bili že identificirani številni tarčni organi, kamor se ta os vpleta, tudi reproduktivno os z jajčniki in endometrijem. Tako da smo postavili nek nov simbol na zemljevid te pokrajine. S tem se jaz zahvaljujem za priložnost predstavitve in sem pripravljena na vprašanja, če se je kakšno porodilo.
Hvala za vašo predstavitev. Zdaj pa povabila Jureta in Tomaža. Aha, Tomaž se že javlja, tako da bom kar dala besedo. Izvoli.
Jaz bi kar postavil prvo vprašanje, mogoče pričakovano. Se pravi, jaz ste že kaj razmišljali, kako bi se pravi vaše ugotovitve spravili na večji, se pravi na neko, da bi bila to neka uveljavljena praksa zdravljenja, kako razumete, to v bistvu so zdaj bile na ravni kliničnih študij, ne?
Ja, tukaj zdaj večina je na ravni predkliničnih, pa nekaj teh pilotnih kliničnih študij, večinoma res iz naše skupine. V bistvu po objavi tega preglednega članka so nas pa kontaktirali letos, zdaj iz globalnih vodstev farmacevtskih podjetij, ki izdelujejo ta zdravila, in so zainteresirani, da se zastavi multicentrične študije, kjer bi vključevali centre, predvsem iz severne, nekaj tudi iz Južne Amerike, pa nekaj centrov v Evropi, tako da bi naredili nek multicentrični projekt, kjer bi mogli vključiti seveda zelo veliko žensk, pa hkrati postaviti te najbolj trdne outcome, ne, se pravi, to so pa živorojeni novorojenčki, ne. Zdaj so te piloti, ampak mehanizem je dovolj verjeten, da so to prepoznali globalno kot projekt, v katerega se splača vlagati naprej.
Hvala.
Mogoče samo še eno malo bolj generalno vprašanje. Zdaj, ko jaz razumem, vi že 10 let približno razvijate na tem področju, pa še niste pri koncu. Pa me zanima, kako gledate na intelektualno lastnino. Zdaj, če bo to zanimivo ratrati za neka farmacevtska podjetja in tako naprej, kaj razmišljate o tem, kako to obvladujete?
S tem pa zdravniki nimamo v bistvu nobenih izkušenj, noben nas na to ne pripravi, nas nauči, tako da pri nas to deluje na tem, raziščeš, publiciraš in potem to je za delitno, da se karkoli naprej razvija. Ker mi zdaj en center nikakor ni v stanju, ne, da bi v bistvu ta koncept dokazal do te mere, da bi bil to res prenos možen v klinično prakso, tako da zdaj, če dodamo nek kamenček v mozaiku, pa da odpremo nova vprašanja, nas že to čisto osrečuje, no.
Ja, seveda razumem. Hvala.
Hvala, profesor Jenštrle-Sever. Zdaj bi pa vabila predstaviti profesor dr. Ines Mandić-Mulec, Biotehniške fakultete.
Pozdravljeni. Rabim pa, da gospa Jenštrle da v stran svojo predstavitev, ker drugače ne morem jaz. Hvala lepa. A se vidi?
Ja, ful se vidi.
Ja. Jaz sem nekaj povedala o bakterijah, se pravi, zdaj gremo od človeka na zelo majhne organizme, mikroorganizme in sicer bakterije. In vprašanja, s katerimi se mi ukvarjamo, so v bistvu vprašanja interakcij med temi bakterijami. In vemo, da v človeških skupnostih, pa v živalskih skupnostih in celo rastlinskih skupnostih, mi vemo, da pride do različnih interakcij in da nekatere so kompetitivne. Lahko bi angleško govorila, sem pozabila, da lahko angleško. So maybe I'll talk in English. I forgot that I can talk in English. Maybe more people will understand me. So in human societies and other societies, we know that members of these societies interact, they compete, cooperate, and even exploit each other. And similar things we are discovering are also occurring in microbial communities. In our research group, we are focusing on social interactions of bacteria and to discover what happens between these bacteria and whether relatedness matter for these interactions, we approach this problem by isolating bacteria of the same species from soil. And here the interest was to really isolate, grouped a collection of bacteria from very small environments with the hypothesis that these bacteria have already interacted in nature. Now, why would one think that relatedness matters in interactions? This is based on theory which suggests that actually in human societies, for example, highly related individuals will tend to cooperate with their relatives more frequently than with those that are not related. And these kind of behaviors are also have been also detected and explored in animal kingdom and as well as among plants. But what about bacteria? So we asked this question and we used to test this hypothesis an assay in which we allowed bacterial strains or isolates to swarm toward each other, and when they meet, they interact, and we wanted to understand this interaction. So here you see a swarm of an agar plate on which bacteria have been allowed to swarm, and they were inoculated on this agar at different points. And you can see that these two swarms, they merge together, while this one does not, it forms a boundary. So has this anything to do with relatedness? Is this nice, kind interaction connected to relatedness? And we confirm that it indeed this is the case. And then highly related bacterial isolates, we call them kin, they merge, and the isolates with lower relatedness, so-called non-kin, form these boundaries. And we predicted that this boundary actually means antagonistic interactions. So we went on and tested these predictions, first addressing the molecular mechanisms between these bacterial interactions. And we found that indeed bacteria encode genes that make them do these differential behaviors. And so if we inactivate these genes in bacteria, this bacterium will then interact antagonistically with its parental strain. So these loci, these genes, were in the group of antimicrobials and toxins. These are bacterial antimicrobials and toxins. They also encoded cell surface molecules and some regulators, probably that were involved in stress response and also killing or resistance to this killing. So we then show in recent publication that actually in this boundary that we see as the consequence of non-kin interactions, the cells are they are like empty sacs, indicating that they die. And in kin merging area, they look quite healthy. The interactions between non-kin in this area also brings stress response, which is here shown as the green fluorescence. And this green fluorescence is the consequence of recombinant strains that we use in our studies that carry these fluorescent proteins regulated by specific promoters that we are interested in. And this particular one is a stress response promoter. So why would we study these interactions, and how could we apply them maybe I can conclude my talk here. So what are the application areas of such a knowledge? Now, we know that bacteria live in communities and that they interact continuously. And these kind of interactions are very important for our health and health of animals and plants. Why? Because it is important how the microbiomes are, so the microbes that live with us. This is one area of basic knowledge, but another one is that we today apply probiotics, and we want to develop good probiotics either for humans that can help us with our health, also in animals, or maybe, and as well as we apply strains, bacterial strains, to promote plant growth or protect plants from disease. Interactions might be also important in bioreactors, where we try to establish a very stable biotechnological process. So what I explained today, I think it's about at the very early stage in a sense of application. This means kin discrimination. So this differential behavior towards each other in the bacterial world in the sense of relatedness. But in the process of this research, we have also discovered some strains that have the potential to develop, to be developed as probiotics. So if I conclude, knowing interactions between strains is highly important when one wants to design a probiotic or a bioprocess composed of different bacteria. And this knowledge is then can be applied also in this development of bacterial groups as products. So I would like to thank you for your listening, and also thank my group, my colleagues who participated in this research over the years.
Professor Mandić-Mulec, thank you for your presentation. So from what you explained to us, this is some new behavior, some new observation, which cannot be, for example, protected with intellectual property rights. So my advice would be, if this is really something that has a market potential, you should keep it as a bigger business secret, if you are planning to work on a product development.
Yeah, I mean, this is the fact that we discovered kin discrimination in bacteria, specifically in very important industrial bacterial Bacillus subtilis, is just the new knowledge, and it is an important new knowledge. Mm-hmm. How can it be applied? I mean, this has not been patented. This is something we think about. Mm-hmm. But during this research, we have also discovered interesting strains that we have already applied patent applications for. And they can act as probiotics, as single strains, not in combination with others.
Mm-hmm. Thank you. Jura? You want to ask something?
Yeah, maybe. I'm just curious, what will be your next step? I mean, I think, what is, you show us some applications here, and where you're going further. What will be your work next year, 2 years?
Yeah, we have already an ongoing project, actually with NIP, with Christina Gruden, where we study this interaction in the context of plants and want to understand how this interaction affects plants. My wish is also to address these interactions maybe with animals more, but this is much more difficult. So these are my future but at the moment, we are when it comes to kin discrimination, we are focusing on plants. When it comes to probiotics, we are focusing on animals, but here not necessarily in connection with kin discrimination at this point of time.
Thank you.
So if there are no other questions or comments, I would like to thank again our speaker, and our next speaker is Professor Dr. Nataša Debeljak. Will the presentation be in Slovene or in English? So I know it will be in Slovene. So it's about kanabinoidni receptorji in zdravljenje hormonsko odvisnega raka dojke. Profesor Debeljak, izvolite. Prosim, vklopite mikrofon, ste mutani.
Se slišimo?
Ja, zdaj bo pa.
No, super. Lepo pozdravljeni vsi skupaj. Torej, predstavitev je pripravljena v slovenščini, ampak z Lukom, sodelavcem, ki jo bova predstavila, bova govorila v angleščini. So, hello to everybody again. So today I will present you the project that we're making in collaboration with Institute of Oncology. So it's about the role of cannabinoid receptors and the treatment of hormone-dependent breast cancer. And this is actually a collaboration between two research groups, the research group from preclinics, from where I come, and the research group from the clinics. So Luka Dobudivishek is a medical doctor that will share and present the presentation together with me. Do you see the presentation?
Yes.
Okay, perfect. So let's see what is the clinical role of this research. There are many patients in Slovenia and also around the world that takes cannabinoids during the oncology treatment. And at the moment, it is not clear what is the role of the cannabinoids on the cancer cells. Do they have pro-tumor or anti-tumor effects? Otherwise, beside that, it is not known if cannabinoids can interact with the standard oncology treatment. And the third thing that is not known is what is the level of the expression of cannabinoid receptors in cancer. As the breast cancer is the most common cancer in females in Slovenia and also worldwide, the focus of our research was about exploring the role of cannabinoids in breast cancer cells. Please, Luka.
So cannabinoids can be divided into endogenous cannabinoids, which are produced by the human body, and the exogenous cannabinoids, which are produced in the nature or are synthetic. Natural occurring cannabinoids are, for example, THC and CBD, which occur in cannabis sativa, or synthetic. We also have synthetic cannabinoids. An example of this is dronabinol, which is a synthetic THC analog, which is currently already used in clinical practice at the Oncology Institute in Slovenia in palliative treatment. So how do cannabinoids work in the human body? They're part of the so-called endocannabinoid system. And the endocannabinoid system has two parts: endogenous cannabinoids and cannabinoid receptors. The main ones are cannabinoid receptors type 1 and cannabinoid receptors type 2. They signal through many secondary messengers and intracellular pathways. So what is their role in cancer is currently not clear. So one other potential problem or opportunity is how cannabinoids affect the standard oncological treatment, which is in hormone receptor positive breast cancer, is endocrine therapy. So there are many possible interactions between cannabinoids like THC, CBD, or endogenous cannabinoids like anandamide with standard endocrine therapy, or even targeted therapy in hormone receptor positive breast cancer. And that is because the estrogen system and the endocannabinoid system are connected on many levels. So just to show you some possible interactions, tamoxifen, which is a standard endocrine therapy in hormone receptor positive breast cancer, acts on estrogen receptors, alpha and beta, which is its primary target and why we use this drug. But it also acts on both cannabinoid receptor type 1 and type 2 receptors as inverse agonist. What does that mean? Well, we don't know. But this interaction, it happens in clinical practice many times for sure. Not because we prescribe cannabinoids, but because patients take them on their own. Other such interaction is the fact that THC also acts on estrogen receptor beta, which could influence proliferation of cancer cells, especially hormone receptor positive breast cancer cells. So our research team is at this point, among other things, also focused on immunohistochemistry, so we can analyze the expression levels of cannabinoid receptors type 1 and type 2 in breast cancer samples, so we can just see what's happening with the endocannabinoid system in hormone receptor positive breast cancer and how it is pathological or physiological role it can be.
So the research that we presented today is actually a collaboration between preclinical and clinical groups. So there are 3 institutes from medical faculty, University of Ljubljana, taking part in it. There are 3 departments of Oncological Institute, Ljubljana, and also National Institute of Biology. Then if you look at the way how we can further explore this research, actually knowing the level of cannabinoid expression, the expression of cannabinoid receptors in breast cancer can further determine the prognostic significance of these receptors in breast cancer. And knowing that, we can then further explore the role of using of cannabinoids in cancer treatment. Of course, on the cell models for the moment. The technology level, we believe that's in the level 4. So we tested the expression level in the clinical laboratory, however, only on a few samples from different cancer, biological samples from different type of cancers. So we have to confirm the expression in more samples. And the commercialization of this project is still in preparation. So thank you. That's all from our side. So any questions are welcome.
Professor Debeljak and Dr. Dobišek, thank you for the really interesting presentation. I right now have a question because I see that you are already thinking ahead. How do you plan to commercialize the project?
Can I? We actually don't at this moment, so it's very difficult. Like this is also but you know there is a. It's not necessarily the commercial impact can be like, you know, there is a giant market for CBD or some or cannabinoids out there, but we are primarily concerned with the safety of our patients, right? If we can say you can take this and there are no consequences for your cancer treatment or your cancer prognosis, it's something else than if we say don't take it, right? Because we are scared that this will impact your treatment, right? So it's for clinical point of view, it's primarily safety concern here, but it's happening every day. So if we discover that if there are no consequences of taking cannabinoids in breast cancer, well, I'm sure somebody will earn more money selling CBD or something like that. But for us, it's difficult to really to monetize this.
Yeah, but you will contribute to the common good.
Yes, yes.
It's important.
Yeah.
I think knowing more about the expression of cannabinoid receptor breast cancer can give us how very important are these receptors in development of cancer, and also how can they interfere with treatment. So I think having a tool to actually detect the level of expression will enable us to do some further research on preclinical and clinical level, then to answer patients, is it safe or is it not safe to use cannabinoids? Because the usage of cannabinoids for the moment, it's quite common. And for the moment, we are very restricted regarding saying, yes, we should say, no, it's not safe till we say it's safe, you know? So just be restricted. So Luca, go.
Yes, if I may add, there is also the possibility that endocannabinoid system is strongly implicated in cancer, and then the cannabinoid receptor expression levels with immunohistochemistry become important. At the moment, there are many antibodies on the market, but none of them standardized, like for sure that we can use like in clinical practice. So it's all experimental. But if it would be important in pathogenesis of the cancer, then it would become important which antibody is the best, and to use that antibody to determine the expression. That's if we like have a good imagination for the future.
Yeah, so this could be another marker for cancer determination, and also for the selection of treatment, you know. So we see that it has potential, but yeah, we are still in the preclinical phase of the research.
Interesting. So I hope you get some good results in your further investigation, which you share with us again. Thank you once again, Professor Debeljak and Dr. Dubovišenk, and I propose we move to our next speaker, Assistant Professor Dr. Maja Andrić.
Okay, thank you. Okay, thank you very much. We would like to present the results of multidisciplinary research, which took place in cooperation between ZRC SAZU, University of Ljubljana, Professor Andrej Šmuc, and Jožef Stefan Institute, Professor Nives Ugrinc. And we studied long-term changes of the vegetation, erosion, and earthquake history in the Julian Alps around Lake Bohinj, in order to better understand how the vegetation and the environment were altered by human activities and climatic fluctuation. And we cored 12 meters deep core, sedimentary core in the lake, and we dated this core. And then geological research was carried out, including sedimentological, mineralogical, and geochemical analysis. Then we also studied fossil pollen grains to reconstruct long-term changes of the vegetation, and also good information about past human activities in the past millennia as archaeological research of archaeological settlements in the area. And roughly about the results. This 12-meter core, in top 4.5 meters of this core, there is a sediment which deposited in the last 6,600 years. And we also detected in this top part of the core, we also detected layers of which were probably formed by earthquakes. We detected 29 earthquakes recorded in the last 6,600 years, with one very strong earthquake about 6,600 years ago, which triggered mass movement of the sediment. Actually, below 4.5 meters depth, we found 4 or nearly 5 meters of mixed sediment. So it was probably quite strong earthquake. And we also detected these specific layers marked in white on this diagram, which probably formed in periods of wetter climate and flood events. And these climatic fluctuations affected hydrology and also geochemical composition of these layers, because deposited in Lake Bohinj, because in these layers there is more flysch material. Today, most of the material comes to Lake Bohinj from the western part of the lake catchment, more carbonate material, but in the past, probably in periods of wetter climates, flood events, or maybe even Lake Bohinj was bigger, some material was coming from the east. So climatic fluctuations affected the chemical composition of the lake. Also, we reconstructed changes of the vegetation, especially forest composition, which showed that we detected significant human impact on the alpine ecosystems for at least in the last 3,500 years since the Bronze Age, when due to grazing in the area, there was a decline of because it's sensitive to grazing, and there are a lot of Bronze Age archaeological sites in the mountains around Lake Bohinj. It was vegetation composition was also affected by iron working, especially in the period between 16th and 19th century, when a lot of beech was cut due to charcoal production for this iron furnaces. Human activities in some periods were very strong and also affected natural processes. For example, 2,600 years ago in the Iron Age, there was a strong forest clearance, a period of very strong forest clearance, and also which probably in combination with wet climate triggered soil erosion and very fast sedimentation rate. And in this time period, the area lost almost all its fertile soil in the region. Therefore, in the later centuries and millennia, these erosion processes were not as intensive as in the past, maybe because there was not much material left to be sedimented. And this shows that erosion processes in the past affect catchment stability also in the next centuries and millennia. Frankly, this is basic research. It's not applied research project, but it has some application potential for application. Especially with this research, we can better understand natural processes, long-term natural processes, and to better understand present-day landscape environment, and also plant protection and management of natural and cultural heritage, and renaturation of, for example, wetlands, lakes, and we can better understand how the landscape was in the past, how it looks like now, and what changes we can expect in the future with climate change or different human pressure on the environment. We can also contribute to tourist industry with public lectures, ecological, botanical, geological paths, exhibitions, museums, education, and also studies of past earthquake records can help us to plan future protection measures. Thank you.
Thank you, Professor Andrić, for this excellent presentation. Are there some comments, questions from the audience?
So I have a question. So you mentioned the tourism, maybe. So do you cooperate with, I don't know, municipality of Bohinj, and there are some plans to present your findings around the lake or something? What are your
not yet. I mean, we occasionally we have some public lectures to present our work, but we didn't go any further.
Okay, thank you.
My suggestion would be, I mean, I find it really interesting, and just join together with Triglav National Park. They have a wonderful visitor center, and they're really, really keen and happy to host you. Maybe you can get your room in their exhibition or something. I think it is something, I mean, today we are not talking about transfer technology, but also to promote the science to the general public. And for this Bohinj part, I think it will be a wonderful contribution if you go for it.
What do you suggest in terms of projects, or how to start?
I mean, just call them. Just call Triglav National Park Visitor Center and say what you have done and what you have offered. They're doing different kind of presentation for the tourists, for the people there, and so on. So I just can imagine your work there, actually.
Okay, thank you.
If there are no other questions from the audience, I would like to thank again Dr. Andrić.
Thank you.
And now I invite Dr. Matej Huš, who will explain to us how bifunctional catalysts work. Dr. Husch, are you with us?
I don't see him. Welcome. Maybe we can continue with next, and if he comes, then we can.
I agree.
Okay.
So our next speaker is Dr. Polona Kugalšek, and she will explain to us the development of a procedure for testing protective masks.
Yeah. Hi. Do you see my screen?
Yes.
Okay, thank you. So yes, today I will show you how we developed the procedure of testing of masks, especially the filtration efficiency of masks. You all remember last year when we were facing one of the biggest health crises that is still lasting, and one of the challenges actually was that we did not have enough of the protection equipment, especially masks. And I guess you remember that lots of different masks and materials came on the market at that time from different materials and also from different parts of the world. And then we were starting to ask ourselves, do we know if those masks are okay? Are they really efficient? Do they protect us efficiently? Are they safe? So do they actually fulfill the requirements of the European standards? And also one of the important questions was whether do really efficiently filtrate also viruses. So in that time, there was quite some questions also coming to our institute from the government, from the ministries, as also from the companies, whether at our institute we can help somehow and test the masks, evaluate them, and so on. So we started to talk with Lotrič Meroslovje company, and we established a really nice collaboration and set up the protocol. Firstly, we set up the device to how to measure, how to evaluate the filtration efficiency, the bacterial filtration efficiency of masks. So we prepared this protocol in line with the standard, with the European standard. Here we are working with bacteria Staphylococcus aureus, which we in the first step, if you see my mouse, in the first step we nebulize or aerosolize. And during this procedure, this machine is the homemade by Lotrič. And this first procedure, we form really small droplets of average size of 3 micrometers. And then those small droplets with bacteria are pumped through the mask. And those who go through the mask are then caught on the plates, on agar plates, which are set in this 6-stage Anderson sampler. After incubation overnight, we then count the number of the bacteria that went through the mask and calculate the percentage of those bacteria, which means that the higher, we calculate the percentage of the bacteria that were caught on the mask. So the higher the percentage, better or more efficient the mask is. With this procedure, which was quite interesting for us, that we were able to set it up in such a short time. We then even collaborated in inter-laboratory comparison that was organized last year, and we actually proved that our protocol is okay. It's in line with the European standard. So we are capable of doing this analysis, also offering it as a service, because we are working in line with the standard, even though that we are not accredited or certified for this protocol, but it is working as it should. So in this period, we tested over 50 different masks, materials, filters for masks, and so on. So then there was still a question, what was with the viruses? What's the virus filtration efficiency of masks? For that purpose, we use the same device that was built, and we just changed the organism. We worked with 2 different viruses, bacteriophages, which are bacterial viruses, MS2 and Phi6. Especially Phi6 is interesting and important in these days because on outer surface or from outside, it's very similar to coronavirus. It has this lipid envelope and this spike proteins, and is also used in different studies as a surrogate or as a model virus for coronavirus. Well, as I said, we set up the protocol. We even tested different masks with this protocol, and we showed that, which was quite an important finding, that the results, when you compare to the bacterial filtration efficiency and viral filtration efficiency of masks, the results are more or less the same, which means that masks, which are okay, which are now on the market, are okay also for the protection against viral spread, because if we really think about what is the main role of the mask, is that it catches the droplets that we are breathing or when we are speaking, that they are formed. So it doesn't matter what's in the droplets; it's the droplet itself that is actually caught on the mask. So yeah, the protocols, the procedures are quite ready. For the bacterial filtration efficiency, I think I can say that we are now already signing or preparing to sign the license agreement with our collaborators, Lotrich, Miroslavia. Similar with viral filtration efficiency, the protocol is ready. Maybe we just need to maybe test a bit more commercial masks to really show that how the protocol is working on huge amount of masks. So yeah, for viral filtration efficiency, we're maybe looking for a company or laboratory that would be interested to establish this protocol in their lab. But yeah, I didn't say that before. The viral filtration efficiency is actually not prescribed in any standard. And it's done in some laboratories, but last year it was only 1 lab in the world that was doing this analysis. So maybe now with this corona epidemic, maybe more laboratories would be interested to have this protocol. So yeah, on the end, I would just like to thank my colleagues who really gave lots of energy last year to establish this protocol. The program, RLS program, was our source of finances. And of course, big thank to Lotrich Metrology Company for really open and close collaboration that led us to the point where we are now. And thank you.
Thank you, Dr. Kugalšek, for this excellent presentation. Are there some comments? Jure, Tomaž?
Yeah, it's nice to hear that we have such institutes that in the time of crisis there are some important impacts and reactions. So maybe my question: do I understand correctly that you developed new procedure and now this will be taken over by Lotrich?
Yeah.
So you will not offer it to other institutes, or you plan to offer like a non-exclusive license to more such companies?
No, for the bacterial filtration efficiency, it's offered, or it will be taken by Lotrich, because we did it together. We developed it together. But for the viral filtration efficiency, we are open to other interested companies or labs. Yeah.
So is this somehow protected? Is it like you have secret recipe, or is you have like you file patent application? I don't know. What's your plan?
Yeah, well, the protocol was developed really quite following the standard, because standard prescribes how this has to be done and so on. But as you may know, standards are never really telling everything what has to be done to achieve this. So there are some secrets, if I may say, that will be now given to Lotrich, so that they will know how to do it. But I think we are not going to patent it because it's something that is prescribed in the standard. I don't know if it's really solid to patent something. I don't know.
Okay. Welcome, Durami. If I may, another question. So you mentioned that you offer this as a service, you know. So maybe it's, from what I heard, it's performing services at the public research organizations. It's something not common, I would say. So there are some difficulties, you know, managing contracting part. So do you think you could do this in the morning? There was discussion also about the big infrastructure. How can we offer this to companies? Do you think you could offer services, I mean, on the large scale also for other services you can perform, or you already do that?
Yeah, well, yeah, we did that, and we also do it on different levels. We offer separate services for on other topics quite a lot, and yes, to one extent it can be done. It was done on our institute. Really lots of work was, or let's say few people were actually engaged to offer service to some big companies. But I think in 5 years, when you were doing something else, not these masks, we came to the plateau because we are not specialized, we are not commercially, we are researchers, so we cannot maybe sell our service so good as it would done if it would be somewhere packed in a company. And also we need to publish papers that cannot be done if you are working as a service provider, let's say. It can be done, but to some extent. So it's a nice maybe side job to have some income, to meet more people, and even with this service, during providing service, you also learn a lot and start some new collaborations for projects and so on. So it's a circle, but to a certain extent, I would say. I think Jure would agree.
Thank you.
Yeah, absolutely. Yeah. Then we go for a spin out at a certain point. And the same goes with this procedure. We will just license it, and we believe that the company will do much better business on it, and we will cooperate. I mean, it's not the end of the friendship on the research based. Absolutely.
Yeah.
Well, I see you have excellent plans for the future, so I'm sure this will be a success story. Dr. Kogaušek, thank you again. And now we move to our next speaker, to Assistant Professor Aleš Lapanja, who will tell you about living coatings.
Thank you very much. So I will just share. Okay, if you please unshare your presentation, please. Can you hear me?
Yeah, I can hear you. Mm-hmm.
Just the presentation of previous. Yeah. Okay. Thank you. Just a second. Okay. Can you see?
Yes.
Okay. Perfect. Okay. Thank you very much for invitation for this meeting. So today I would like to speak about the procedure, how we develop the living coating. This is one of the things that we are developing in the College Biology Laboratory at the GSI, and also one of the, let's say, things besides many others, how our, let's say, that can be seen how our approach is actually can be very useful, and it's kind of switching in the understanding of let's say in this case, anti-corrosion. So we all know that the corrosion is a very big problem. So either it affects all kind of infrastructure, or it can affect the traffic by destroying boats. Well, what we can see here in these cases is just more or less physicochemical corrosion effect. But besides this physicochemical corrosion, we have also biological corrosion or biologically promoted corrosion, which is, let's say, a result of interaction of different sorts of microorganisms and their metabolism, and how this then starts to dissolve the structure. So in most cases, we are thinking in the way that we would like to avoid bacteria to form biofilms on surfaces, either by repelling or killing them. In one case, which is well known, it's like exclusion, steric repulsion, electrostatic repulsion, low surface energy, so to change the surfaces, or to actually use biocides, or in any case, in some cases, contact active biocidal effects. Well, at the end, if you see some cases, for instance, here, that the bacteria present on the top of these, let's say, structured surfaces, even they die, they are actually forming new film on the surfaces. So this new film on surfaces has nothing to do with the previously very nicely structured surfaces, and it is very good environment to establish new biofilms, so the new microbes can be attached on these dead bodies of these dead cells here on what we can see. So what we were thinking, so we're thinking, okay, if the biofilms are inevitable part of our life, we need to interfere how, or to manage how these biofilms are going to be formed. And we have a good example here on the right, that in, let's say, in the biofilm formation in our mouth is actually quite, let's say, established process by multiple steps, and for each step must be preconditioned by other microorganisms. So like in the first, at the beginning, we have some streptococci, and then following all to the top kind of structure. So we were thinking maybe we can apply quite similar system to attach different microbes to make biofilm and to exclude the others that can promote corrosion, as we can see in the left side. And this is the question how to make that. So we started from the beginning, from the basics of the let's say, the basic property of the cell, which is negatively charged. And from that, we could then make very thin nanolayer structures on the cells, and which actually change the, this approach actually changed the surface charge. By changing the surface charge, it's very simple then to manipulate and to make either the biofilms on the surfaces, or to make aggregation of different bacteria species. So when we had this tool ready, so then we started to think about how to make very nice structure. So in this case, what I'm presenting here, we actually, firstly, this is on the bottom, we see the scheme, how the, let's say, the metal is then, on the metal we deposited cells. And the first layer of cells are actually cells that are more or less dead, but they accumulated some antimicrobial agents. And the second layer is actually elastomer, which prevents the physicochemical corrosion. And over that, we have layers of bacteria which are alive, and they produce or exclude other microbes that can grow in that. So after we had this structure and made this in the labs, then we started to make upscaling and to make easy coating system. So we actually used the deep coating system, where we actually layered these layers of microbes on the top, and on the right you can see how this microbe, one of cell, is actually attached in very nice surface. So after deployment in the sea, in the real situation, these coupons, we actually took it into the lab, and then we checked with the metagenomic tools how the community was developed. So on the left side, we have two controls where more we have colored structure, more communities or more different species or genera are present, which are affecting the biological corrosion. So actually these are biocorrosive bacteria. And when we cover with the elastomer, which is just chemical prevention, we still keep quite some bacteria which are promoting corrosion. So if we have mechanical scratch on the surface, then immediately we'll start the corrosion process. And in our approach using artificial biofilm, there were no bacteria which can promote corrosion. And now you can guess which is protected by living coating. So I will not tell, which is obvious. And thank you very much for your listening. And these are our, let's say, financing aids that promoted our work, and we can do with that. Yeah. Thank you very much.
Thank you, Assistant Professor Lapanja, for this really excellent presentation. I think this has a wide range of application possibilities, and you should work further on this. So my question is, do you already have some interaction with the industry, with some partners who would be interested in further developing this innovation?
Exactly on this, not yet. We have another, let's say, another activities which are, let's say, not so similar, but in the concept, let's say, similar. But I would like to say that the approach is maybe a little bit too advanced at that point. So the industry is not ready how to apply or what to use and what sort of extent is needed. And we are now thinking into how to, let's say, either to decrease cost, to use some waste material to coat cells, which we had some nice results currently, to, let's say, to attract the industry, or let's say trying to promote this sort of, as I'm here showing what we can do. So so this is quite, let's say, a new approach. Yeah.
Mm-hmm. Are there some other questions, comments from audience?
Just maybe I can mention also that this approach we are also now applying into the prevention of the tooth deterioration, so that the processes that are happening in our mouth by using probiotic microbes, and we are also quite efficient in that. Yeah. Yeah. Okay.
Thank you once again. And now we come to our last presentation, which will be given by Assistant Professor Dr. Andrea Ramšak. The floor is yours.
Thank you. Good afternoon. Okay, I think you can see my presentation.
Yes.
So, the presentation is based on a recently finished project, which was financed by RRS and the Ministry of Agriculture. And the main aim of the project was to make an overview on Slovenian seafood market, and also developed some tools which are based on DNA technologies. Some tools are already developed, but not so much. So food security is one of the main issues, and as well seafood as well, because the seafood is one of the most traded commodity on the world. And what is interesting is that we have almost 2,000 species of fish, cephalopods, crustaceans, and algae, which are registered in FDA as fishery products. And in this presentation, I will focus only on squid species. And we know currently 180 squid species, but among them are 30 to 40 are economic important squid species. The topic is also relevant because of the fishery and exploitation of natural resources, because of the high demand for seafood. This makes a huge pressure on fishery and also pushed illegal and unreported fishery. And according to the estimation, 25% of fishing is illegal. And because of this, it also has implication for the fishery industry and also for the health of the customers. Customers and have a right to know what to eat. So the EU have regulation in which proposed which basic information must be available for consumers. Here we have a comparison between Ireland and Slovenia, and we see that at the Slovenian fish market, the labeling is very good, even though the labeling and traceability is one of the main issues in the fishery industry. This is usually related to adulteration. That means that goes to species substitution, let's say substitution with Asian catfish, which is very cheap and not valuable fish, is substituted as grouper, or other fishes, let's say panga, is also substituted with other much more valuable species. Then the problem can be undeclared product extension, and they also put raw material into other processed fish products. Then the big problem is provenance, so fishery substitution. So we don't know the geographic origin of the products, and especially abuse of custody. And then also ethical issues, because we have as well practices of illegal catch methods and even modern slavery at the vessels. So here I will present what we do with squids. So firstly, we search the Slovenian market. So we're sampling in the 10 Slovenian cities in small and big shops. And the first thing which we are faced that we cannot use classic taxonomic identification, because the squids have a quite low number of useful taxonomic identification, and usually they are, when they're sold, they are without skin, or also only the body, and so on. So we can help with the genetic authentication of species. So we have some classical methodology available, or tools like PCR coupled with RFLP. Then one of the methods useful is also barcoding. Usually people use 16S/CO, cytochrome B, or ITS regions to make sequences and then compare them, which is quite long procedure, quite demanding. Then we have some qPCR tools, also some LAMP design. But when you put together, we have only tools or assays for less than 10 species. And that's a problem. So we start our study with tissue sampling, and then we proceed with barcoding, because we would like to know very precisely which species appears at the Slovenian market. So after the barcoding results, so we identify which species we have on the basis of CO1 sequence analysis. We also did intensive and extensive phylogenetic, bioinformatics and phylogenetic analysis of the CO1 marker. Why? Because the biology of the squids are not very well known. And we have to be sure that we can use those markers, and that we don't have any troubles with different biological processes like hybridization or heteroplasmy or so on, let's say, because this can be in some species a great problem. So after this intensive bioinformatics work, we were then ready to design a qPCR assay for Loligo vulgaris, which is one of the very common species in the Slovenian market. And it's also abused because it's usually declared as an Adriatic squid, which is actually not a right name, and it should be sold under a European squid. So what are the results? So then we compare our results also with how they are compliant with seafood list, which is valid in Slovenia. And we found that slightly more than half of the samples have Slovenian commercial name. Less than 50% of the samples have scientific name on the declaration. Some of them, half of them, have also the geographic origin. And when we check the results of barcoding, we actually found that that 41% of the samples are compliant with the seafood list. So that means that what is on the declaration, it's also this is the right species. And this is quite a bad result. There are several problems because also the commercial names and seafood list, which is available in Slovenia, is not updated. And these results also pushed us further to design a new tool, as I mentioned previously. So we designed a qPCR tool to identify Loligo vulgaris. As I mentioned as well, some there are problematic because seafood is not updated. And also here is one example, which is example of Mitre squid and Indian squid. These are two species which has overlapping areal in the Indian Ocean and Pacific Ocean. And they are actually, they are actually not in the seafood list, and they are imported in Slovenia under the name of Loligo hinensis or Loligo duvaseli. And we found by the end that we have actually totally different species at our market, which is Uroteuthis duvaseli. And we found that the most common species is Patagonian squid. And usually Patagonian squid is sold under different names and usually is substituted under the name of Adriatic squid, which is Loligo vulgaris. Why is this? Because Adriatic squid or Loligo vulgaris has much higher price than other species which are available. Also because it has much better quality of meat. So as I said, we were pushed to design a new tool, and new tools has to be simple, has to be very reliable, has to be very simple to use because there are a lot of application areas which needs those tools, especially in Slovenia. They desire to have very simple tool at the custom because, as I mentioned, classical identification methods are not possible, so they need a fast and efficient tool for species identification. And the application areas are in food processing sector because they want to control raw materials, as I mentioned as well in the problems in the custom abuse. Then also supermarkets and retail stores, they want to check the products. Then fishery management authorities need efficient tools also because they need to know about the stock exploitation, and they need to be sure that the species is identified properly. And in this era of declining of biodiversity, we also need a new tool for biodiversity monitoring and nature conservation as well. So we would like to commercialize the project. So we are looking for funds and partners. We are also looking around for the need in food industry in Slovenia. There are some interest and they would like to have specially designed assays according to their needs. And at the moment, as I mentioned, we have on a seafood list, almost 2,000 species. And at the moment, we have only one assay which has a patent, and it's a really fast, efficient, and simple. And this assay is dedicated for species identification in fish, and it's able, in one run, we can identify only 20 species, and in total, the database have currently only 60 species in the database. So in total, currently we can identify in a simple way only 60 species of fish. So I think that the idea and the project has a lot of, actually a great future, because there is a lot of work and which has to be done so that we can satisfy the need in the fishery and food processing sector. I would like to thank also the partners at the project. This is, beside the National Institute of Biology, is also University of Primorska, Department of Mathematics, Natural Sciences and Information Technologies. Thank you. So I am available for further questions.
Thank you, Assistant Professor Ramschak, for this really great presentation. I see that this innovation has really a big market potential. So I'm wondering, did you take care of any IP protection at this point?
The project was recently finished, so at the moment, no data are released to the public. Yes, we are looking. Yes, we would like to design a commercial tool, but on the other side, we are also would like to offer our knowledge and the assays also to the other sectors. As I mentioned, such tools are also necessary, or at least the bioinformatics information are necessary for biodiversity monitoring. And there are also the great potential to develop slightly different tool, which is much more open with data, while the industry needs something simple and efficient for species identification.
Mm-hmm.
Maybe I also have a question. So have you identified to whom you would sell your knowledge? I mean, would that be like a national laboratory for safe food, or more like private companies that would produce a device for detecting the origin of species? Have you thought about it?
Yeah, at the moment, the much more interest comes from our Slovenian national authorities, and especially from inspection authorities, because they need tools. Yeah, that would be great to offer products as well to private companies to develop it further. Because according to my knowledge, there is only one company in Germany, I forgot how the name, which is the only one company which produce qPCR assays for a very limited number of species, because this is needed because of the health issues, because of the allergies. And then maybe this is also important that in the last 2 years, there are some publicly financed projects which are also trying to develop different tools, but mostly on fish.
Okay, thank you.
Are there some other questions and comments? If not, I would like to thank the speaker once again. And at this point, we came to the conclusion of this session, and I would like to hand over the moderating to the organizer.
Yes, Marietta, please.